So, a very good afternoon to one and all who has joined us in the afternoon session of this 1 week short-term training program organized by Ballarpur Institute of Technology, Ballarpur, and sponsored by All India Council for Technical Education. As we are all aware, the title of this STTP is Biotechnology and its application in tribal agriculture areas. And to address the afternoon session of second day of this STTP, we have with us Dr. Pravin Patil, sir. And I would like to introduce our honorable guest for this session, Dr. Pravin Patil, sir. So, Dr. Pravin Patil is professor in Department of Basic Science and Humanities of Narsimunji Institute of Management Studies, University, Mumbai. He has completed PhD in Green Technology from Institute of Chemical Technology, which was formerly popularly known as UD City, Mumbai. He has completed his PhD under the guidance of Padma Shri Professor Ganapati D. Yadav. Dr. Pravin Patil was also a research associate in translational research for Primal Life Sciences Limited, Mumbai. He has worked as an operator in charge of scanning electron microscopy at Advanced Lab Department of Chemical Engineering, ICT Mumbai. Furthermore, he has also mentored BTech students from NITs and IIT Bhubaneswar during summer internships at Institute of Chemical Technology, Mumbai. He has published more than 30 research articles and also published more than 9 book chapters. He has many certifications in his name that include BIRAC certification on bioentrepreneurship, grant writing, and intellectual property management at IIT Bombay. Another important certification he has passed with 92.4% is in industrial biotechnology course organized by Delft University of Technology, Netherlands. Then he has also completed a certification on laboratory safety organized by TICWIP, that is TEQIP, and ICT Mumbai, and a finishing school come training program certification on preparative processing and analysis of biopharmaceuticals, which was again sponsored by TEQIP, that is TICWIP at ICT Mumbai. Dr. Pravin Patil is a life member of Biotechnology Research Society of India, BRSI, since 2014, and life member of International Bioprocessing Association, which is an international forum on industrial bioprocesses since 2017. He is recipient of Best Elsevier Poster Presentation Award at ICCP 2016, organized by Vellore Institute of Technology, Vellore. He has also been appreciated from JMB Pharmaceuticals Private Limited, Mumbai. And he has also received Shri GM Abhankar Research Paper Presentation Award at International Conference NHBT 2017 in the field of biomass conversion. Dr. Pravin Patil, sir, we are glad that you accepted our invitation and agreed to be available as a resource person. May I now request you to please start the session.
Thank you, Dr. Mishra, for such a kind introduction. I'm eagerly looking forward to this STTP program. And let's dive into the presentation. Let me just give me a second. So, yeah, okay. So is this presentation visible to all of you?
Yes, sir, it is visible.
Yeah, okay. So the title for the presentation is Synthesis of Biofertilizer and Biopesticides from Agricultural Waste Using Biotechnological Tools. This topic will basically comprise the possible ways of obtaining biofertilizers and biopesticides using agriculture waste. So let me give you a brief idea of what is the content to be expected in this presentation. The first part would be the introduction about the current scenario of agriculture sector in India. It will be followed by the current challenges specifically related to the agriculture waste. Then we will look into the biofertilization, biofertilizers and biopesticides, their types and their production processes, synthesis processes. And ultimately, we will discuss the challenges and opportunities involved in the process. So let's go to the first part, which is introduction. Let me give you some, let me share you some some facts about Indian agriculture, that we have almost 328 million hectares of graphical area, from which 142 million hectare area is being sown every year by our farmers, and that is why India is popularly known for its agriculture sector. The gross cropped area is about 190 million hectares. And apart from that, India also hosts more than four diversity hotspots, including Himalayas, Western Ghats, Indo-Burma region, and Sundan Lands. All these hotspots are popular for comprising more than 46,000 species of plants and 86,000 species of animals. So you can see that India is highly enriched with the biodiversity. However, this highly biodiversity also bring a challenge of soil fertility that needs to be addressed considering the current scenario of global warming and pollution. Apart from that, India is also the second largest producer of fruits, vegetables, wheat, rice, groundnut, and sugarcane. So you can imagine, if since we are the second largest producer, the whole resources and the technology should be directed toward the betterment and efficient enhancement of the agriculture sector. Uh, However, there are some challenges, critical challenges that we need to address, considering the current scenario. The first one is the declining crop productivity in India. So, in last 2 decades, India has witnessed a declining crop productivity throughout the nation, and there are several reasons for that. The first one is the imbalanced and indiscriminate use of chemical fertilizers. The excessive and imbalanced use of chemical fertilizers has spoiled the fertility of soil and led to the several pollutants in the environment. Apart from that, as a human, we need several nutrients, right? Like proteins, carbohydrates, etc. Similarly, plants also do some kind of nutrients, some salt, some minerals. So in case of deficiency, their yield would be hampered, and that is why the uh, supply of particular nutrients should be provided in an adequate amount. Apart from that, rain, the two-third area of India is rain dependent agriculture, and that is why we do not have enough water sources. So if there is a drought in a year, then that whole product or the whole yield will be drastically affected. Similarly, the inadequate irrigation practices, along with the continuous fragmentation of land, has led to the unfavorable adaptation of technology. So there are so many things involved in this process. Those are responsible for declining the crop productivity. However, the major concern is still the chemical fertilizers, the excess use of chemical fertilizers. So see so that is why what we are doing is we are trying to, since we are trying to exploit the soil, exploit the land by harvesting more and more crops day by day, what we are doing is we are not allowing soil to get nurtured naturally. And in the process, there are several nutrient deficiency are forming. As you can see, plant needs nutrients that are taken up primarily by the roots in the form of an aqueous solution in the soil and getting transferred to throughout the plant. However, beside NPK, which is nitrogen, phosphorus, and potassium, sulfur, zinc, and calcium are also required in some amount. These nutrients, whether it is micronutrient or whether it is macronutrient, both should be provided in an adequate amount. Other nutrients such as iron, boron, even though they required in a small quantities known as trace element, their deficiency can also cause a severe effect on the yield of the product. However, several results have showed that the significant increase in yield can be obtained when enough amount of micronutrients along with NPKs are provided to the plant. So the one problem we have understood is that the nutrition deficiency in the soil, but that is not all. There is another one. So the other problem is the agricultural waste. So even though there is day by day the production is getting reduced, there is also another problem, which is agriculture waste. So let me put some interesting facts about the agriculture waste in India. India is short by 10 million tons of cold storage capacity. So whatever product we have in our hand, the fresh product cannot be stored. We do not have the facilities. The farmers do not have the facilities, and almost 30% agriculture produce goes waste every year. more than 20% produce from the field is lost to the poor post-harvesting practices. So since our country is developing, we are facing several challenges throughout the agriculture sector where the post-harvesting facilities are not properly well equipped. However, 10% of food grain that is India produce annually is eaten by rodents. So we know we hear this every year that in Punjab several tons of wheat is being spoiled by rodents. So this is very common factor which needs to be addressed. Apart from that, only 7% of food in India is processed. Only 7%. Can you imagine? 93% is being wasted. When we compare that with the United Kingdom, 65%, more than 65% is processed in United Kingdom. And even if we compare with the developing countries like Philippines, they process more than 45%. So we are extremely lacking in this field, and we have to catch up with the pace. So India, even though the India is the largest producer of several things, several products, we waste more than 25% worth of produce every year. So now we have identified there are two major problems. The one is the nutrition deficiency in the soil, and the second is the agriculture waste. So what are the intervention that can be done in this field to sort this out? So there are basically two major things. The one is the cloning of superior genotypes. As we know, if we can modify the genetics, modify the microorganism at a genetic level or plant at a genetic level, we can add some variants, add some strains, add some traits in that particular plant, and make that plant disease resistant or drought resistant or sometimes even pesticide resistant. So this is, but however, there is a huge ethical problem with the cloning of superior genotypes. Then there is another option where we use biofertilizer and biopesticides. So instead of using these chemical fertilizers, we use biofertilizers, which are originated from the biosource. So let's talk a little about the agriculture waste. So annually, Asia alone generates 4.4 billion tons of solid waste and municipal waste, comprising 790 million tons, of which 48 million tons are generated in India. By the year 2047, municipal solid waste generation is in India is expected to reach 300 million tons. This is a huge amount. So all this waste, whether it is coming from the crop yield, whether it is coming from the food processing, all this waste is being dumped in a yards where mostly they are not, only 2 to 3% is being recycled or being processed. So this remaining 90 to 95% must go through some useful application. So considering these two challenges, whether it is declining the crop productivity or whether it is agriculture waste management, why not find a solution which can tackle these both problems? So biofertilizer is a solution where we can also increase the crop yield productivity by using agricultural waste as a source for this biofertilizer. This source could be in a different manner. Either we can use this source as a carrier. What is carrier? I'll come to that part in the next slide, next session. We can also use those biomasses for the production of particular microbes, which are again used as a biofertilizers. So we have to take the problem into account and find a solution. So why everyone is hating this chemical fertilizers? When chemical fertilizers came in the last century, everyone was very excited, and farmers were very happy since they were obtaining multiple folds of profits from their crop yields. However, eventually they realized that these chemical fertilizers, which were being sprayed on the soil, are getting shifted into the water basins, water tracks, and destroying several microorganisms, several microflora that is very essential for the soil fertility. So when soil has number of microbes, this constituum of microbes which work together in a symbiotic association, they offer a very stimulative environment to the plant. However, this excess chemical fertilizers have killed those microbes, and have those microfloras have been vanished from the surrounding, and this has led to the reduced soil fertility. and this overall is causing damage to the overall ecosystem. So recently, in last 1 decade, we are listening to this term, that's organic farming. Actually, organic farming is not new. This is actually what we used to do, which we have lost in the process. Earlier, what we had to do, if you could recollect, when in our childhood, we use, actually I am from a farmer background, so I am aware of this, that when I was a child, what we used to do is we used to had a cow dung and everything as a fertilizer for the crop. No chemical fertilizers were used, and that time the productivity, that thing is known as organic farming, which is trying to get resurface back in this current era of pollution and global warming, which is a good thing, which is a good step. But organic farming cannot be attained without the help of biofertilizers. And to obtain that organic farming, we must understand what is biofertilizers. So basically, when you hear this term biofertilizer, it means a live microbial preparation that can be applied either to the seed, either to the root, or either it could be added to the soil where plants are supposed to be grown. And they start colonizing in the rhizosphere. Rhizosphere is the surrounding area near the roots. That region, they start growing and they start forming a symbiotic association with the roots and start providing the insoluble nutrients, which are present in there, but they are in insoluble form. They start solubilizing them so plant can uptake it. So this basic, that is the reason biofertilizers are also known as microbial inoculants. So these microbial inoculants can help plant obtain the nutrients which are present in the surrounding. So there are different types of biofertilizers such as rhizo fertilizers, azo fertilizers, azo spiridium fertilizers, azola fertilizer. These all nomenclature of fertilizers are on the based on the type of microbes which are being employed in the process. So again, why not chemical fertilizers and why biofertilizers? Let me briefly tell you that also. They are, first of all, they are environment friendly. They are completely biodegradable. No remains will harm the environment. Pose no danger to environment. Advantages over chemical fertilizer in case of economical aspect, even in case of the efficiency aspect. They can replace 25 to 30% of chemical fertilizer, which is a huge thing, I could say, if we could really implement it at a grassroots level. And they can ultimately lead to the productivity of that particular plant. So what is the current market scenario of biofertilizers? Currently, 25% of the requirement of chemical fertilizers can be replaced by biofertilizers. However, the coming demand of biofertilizers is estimated to be 27.3 lakh million tons, but we are only able to produce 0.86 lakh million tons. So you can see the difference. There is a huge gap between requirement and the production. So that needs to be addressed. So now we have understood what is the market of biofertilizers, what are biofertilizers. Let's dive into and try to understand what are the different types of biofertilizers and why different biofertilizer is called that particular name. So as you can see on your screen, uh, if it fixes the nitrogen, the biofertilizer is known as a nitrogen fixer. When it helps solubilizing the phosphate available in the surrounding, it is known as phosphate solubilizing microbe. Similarly for sulfur solubilizing microbe, similarly from plant growth promoting one. Sometimes these biofertilizers just help to uh, enhance the growth of the plant. Similarly, there are several types of fungi involved, which are also subcategorized into root interphytic fungi as well as plant growth promoting fungi. Similarly, they help promoting the growth of the plant. Then there are azola fern. Fern are the vascular plants. They can produce with the help of spore formation. Then in nitrogen fixation and phosphate solubilizing microbes are the major categories. In nitrogen fixer, there are main two categories, bacteria and blue-green algae. The bacteria are again subdivided into the rhizobium, azotobacter. These are the types of different bacterial strain that can be used for the nitrogen fixing agent in biofertilizer sector. Similarly, blue-green algae have different types of algal species that can be also employed in the process. So this is just to give you an idea what kind of biofertilizers can be obtained using microorganisms. These are the images of biofertilizers. You can see Azo Spiridium, phosphate bacteria, azotobacter, rhizobium, blue-green algae, and rhizobium on your screen. These are the examples of the particular strain which can be used as a biofertilizer in that particular application. When we are looking for nitrogen fixing bacteria, biofertilizers, there are free living, symbiotic, and associated symbiotic. Free living microbes are the microbes which they don't care whether it is they are getting something in return or not. They are doing their regular metabolite and producing something which is being helpful to the plant. Symbiotic association is where both are getting benefits from each other. That bacteria is producing something which is beneficial to the plant, and plant is producing something which is beneficial to symbiotic fungi. And the associate symbiotic is another type where they just have a mutual understanding to grow in the same surrounding. However, there is no specific requirement or demand is there. So similarly, Azotobacter, Clostridium, Nostag, these are the type of several bacteria which can be used in nitrogen fixing as a nitrogen fixing biofertilizers. Similarly, several and several aspergillus and penicillium fungal species can be used for the phosphate solubilizing biofertilizers. What they do, they solubilize the phosphate present in the soil, and that insoluble phosphate getting converted into the soluble form, which can be uptaken by the plant. Similarly, the potassium mobilizing biofertilizers are also there, where several types of mycorrhiza and several types of bacterial species can be employed. Then there is biofertilizers for micronutrients. So silicate and zinc soluble. See, micronutrients are the nutrients which are essential but can be provided with the trace amount or small amount. So for that also several bacillus species can be employed. Similarly, for promoting the plant growth, we can employ Pseudomonas fluorescens bacteria for the purpose of growing the plant at a faster pace. So basically, as I told you earlier, the categorization is basically mainly divided into nitrogen fixer and phosphate solubilizing agent. Let's also see how there are other types as well. So there is potassium mobilizing biofertilizer, zinc solubilizing biofertilizers, plant growth promoting biofertilizers, potassium solubilizing biofertilizers, and there is vascular arbuscular endomycorrhiza, which is a fungal species biofertilizers. We'll see one by one briefly. So nitrogen solubilizing biofertilizers, what it does, it works in a two different way. First, it infects the plant root and forms a nodule or a lump, which is known as a nodule, and this promotes the nitrogen fixation at that particular site. However, at the same time, it also produce an enzyme known as nitrogenase, which is efficiently uh, provide the supply of reduced nitrogen to the host plant. So in this manner, in this both way, that nitrogenous rhizobium species is helping to the plant to grow. This is mainly suitable for these types of plants, including groundnut, black gram, red gram, cowpea, etc. Then rhizofertilizers, since rhizobium bacterium is used, they are also known as rhizofertilizers. These are just different names. Please do not get confused. So what happens is here they have this ligmoglobin protein molecule which carries oxygen to the bacteroids. Bacteroids are the uh, rhizobium species structures which are actually found at the site, and this pink color is given by this. That is why you could see these pink color nodules in some plants at their roots. So this is nothing but a rise of fertilizers. Then there is another type which has the same purpose of nitrogen solubilizing agent, is known as Azo Spiridium. It is a bacterium which is um, highly useful to promote a growth of plant particular species. Apart from providing the solubilizing nitrogen, it can also provide several growth promoting factors such as indolysitic acid. As those who have worked in plant biotechnology, they must be knowing that indolysitic acid, gibberellins are the major growth hormones which are involved in the growth of particular plant. So this can be also provided by these plants. This azospirilium is mainly suitable for oil seeds, fruit and vegetables, sugarcane, banana, coconut, and etc. Then these are again specifically specified examples of nitrogen solubilizing biofertilizers. Several azobacter, then clostridium, then some Klesbia, rhizobium, Azo Spiridium. These are the different species. However, they have different way of mechanism. As I told you, what is free living and symbiotic, and what is associative symbiotic. So this is the way all these microbes are helping to solubilize the nitrogen which is present in the soil. Then the second type is phosphorus solubilizing biofertilizers. These are the major nutrient for phosphorus is a major nutrient for plants, and that can be fixed by these specific bacteria, which the most common bacteria which is used is Bacillium magneterium. Those can grow and secrete organic acid which dissolve this unavailable phosphate into soluble form. So whatever insoluble phosphate available in the surrounding is been dissolved with this organic acid which are secreted by this bacillus species and can be used by the plant. It improves the soil quality and subsequent uses ultimately. Again, this is the specific examples of those particular microbes. These are the bacterial strain and these are the fungal strains. Then there is another third type which is known as potassium mobilizing biofertilizer. This all terms are self-explanatory. Just look at them and you will understand. So what it does, it mobilizes the potassium. So mobilizer is a beneficial bacterium capable of mobilizing potassium available in the soil into the root zone of plants. So there are several benefits of using it. So it basically mobilizes the insoluble potassium. Again, the same thing. And this potash mobilized is immediately available to the plant. So that stimulates the flowering and fruiting in several plants. And this is very essential. This is very crucial thing for growth of particular plant. It simultaneously, it also improves the soil properties and sustain soil fertility. So it helps maintain the sustainability of soil fertility. So if continuous use of biofertilizers can, in a long term, can definitely enhance the soil fertility. It also encourages the plumpness and suckleness of fruits and grains and increases the Brix level. Brix level is the level of the amount of sugar present in that particular fruit. So it also help enhance the Brix level of particular plantical product fruits. It also can increase the yield from 10 to 20%, which is efficient. 10 to 20% is quite acceptable. It also improves the quality of produce and then thus fetches the good price. If the products which are farmed, which are produced, have better quality, they can definitely fetch a good price in the market. So these are not just helping the environment, but this can also pay the farmer off with the help of economical benefits. Again, these are the specific examples of potassium mobilizing biofertilizers. These are this is the same scanning electron microscopic image of these biofertilizers. So Bacillus species is commonly used for the as a zinc solubilizing agent, where zinc mobilizer, the pure culture can be so this mobilized zinc, which is mobilized by this particular species, can be again uptaken by the plant. Then there is another type, vesicular aspicular endomycorrhiza, which is commonly known as VAM. This is highly common. 80% of the plant on this planet have chances to have this with them, with on their roots. So this is highly common. So if you see the microscopic structure of them, you can see this hyphae. The hyphae of VAM, what it does, that microscopic tubes can colonize crop roots and grow out into the soil further than the root hairs. So they literally extend the roots artificially and try to uptake the nutrients from the surrounding. So that is where they are commonly found in all kind of plants. They can VAM hyphae also can be help to retain the moisture. As we all know, longer the roots, highly branched the roots, they have more capacity to hold more moisture and water. And even though these are at microscopic level, when you look at the mass culture, they can definitely help hold the moisture for longer time, and that ultimately enhance the fertility of the soil. So there are these WAMs are mostly used for turmeric, banana, rubber, coffee plants, and there are two more. There are two more, and then we'll move to the next part. The blue-green algae. Okay, you can look at the this image. This is the blue-green algae real image. Let try to understand what is it. This is a type of algae, okay, which mostly involved in the nitrogen fixation. However, it also helps in several growth promoting activities. So they are microscopic, okay, they are widely distributed in the aquatic environment. So that green color clumping in the lake that you see is most of the time is brugline algae. And the one thing which is to be noted with this microorganism is that they can survive at extreme temperatures. They can adapt their structure to the more than 85 degrees Celsius. So the regions where the temperature is higher, in those regions, these microbes can be efficiently used since they can survive. These algae can be efficiently used since they can survive at higher temperature. They have symbiotic association with the plant around which they are growing. And what happens is that they have this blue-green algae have the ability to photosynthesize food and fix the atmospheric nitrogen during their symbiotic association. Okay, as we all know, these are autotrophic organisms. So they can create their own energy, right? Blue-green algae can create their own energy. These are autotrophic organisms. So these autotrophic organisms can obtain energy using photosynthesis while fixing the atmospheric nitrogen for the plants. So they have again they have the symbiotic relationship with the plant. Blue-green algae are of immense economic value as they add organic matter to the soil. So when the crop is harvested, this blue-green algae are still there, and they keep growing and keep enhancing the fertility of that particular site. So it is again a very fruitful returns in a long term. Then the last one is the mycorrhiza. This is a fungal type. You can look at this image. You might have seen this type of structure when you can see this white color fungal filamentous structure at the root of several plants. So this mycorrhiza is a symbiotic association between green plant and a fungus. The plant makes organic molecules such as sugars, which are provided to the plant to the fungus as an energy source, and in return, fungus supplies the plant water, mineral nutrients such as phosphorus. So this is basically involved in the phosphorus solubilizing as a phosphorus solubilizing agent. They can apart from that they are drought tolerance. What we understand by the drought tolerance means that they have ability to hold as much as water as possible. So they maintain the water balance, and ultimately they maintain the overall plant growth. So now we have understood what kind of biofertilizers, what kind of microbes can be used in the agriculture sector. Now try to understand how these biofertilizers can be produced with the help of agriculture waste. So let's go to the introduction part, a brief introduction that there are basically 4 types of formulation in biofertilizer. The first one is the liquid formulation. So where the microbes are dissolved in a liquid form that can be directly sprayed to the landfill. Then second is a carrier-based formulation. Try to understand this term carrier. What it does, these structures carry the microbes on them. So what is the need of, so you might be thinking, what is the need of this carrier if we can directly spray or directly spread the biofertilizers on plants? The reason is sometimes the production of biofertilizers and their actual implementation, there must be some time gap in between. So these carriers should able to hold those microbes alive for that particular time. So they can be viable until and unless they are applied in the field. Then there is another type known as granular formulation. This is a granular formulation where granules of these microbes are being prepared, which can be used for the biofertilizer application. And then the last one is the encapsulated formulation, where these nutrients, or sorry, these microbes are freeze dried uh, and used and put into the encapsulated form and can be sprayed as a tablet. So as you can simply, just by looking at this slide, you can simply analyze that the carrier based and liquid formulation should be the economically feasible, right? Even though these are available in the market, these are, the processing cost is way too high, and we would prefer liquid and carrier based formulation. However, there are differences between carrier based and liquid based formulations. So carrier based, so again, let me tell you what, when I say carrier, that means that microbe is carried by something. So it is a vehicle on which that microbe is being carried. So it can be carried from one place to another place. It can be carried from one day to after 1 month. It can be carried to that. So that holding of that particular microbe for that particular time is done with that carrier, and that is why it is known as carrier-based biofertilizers. So advantages of carrier-based fertilizer: they are cheap, easier to produce, and require less investment. So when this STTP is about the development of agriculture in rural field, in tribal areas. So tribal area people do not have uh, sophisticated facilities or high amount of investment. So they must have an option which is feasibly available and can be used immediately in the field. So even though the carrier-based biofertilizer have their advantages, they also do have their several disadvantages, such as they have low shelf life. So that means they won't be, those microbes won't survive for longer time. Okay, so after some time they would die eventually, or they would go into the dormant stage, and that will ultimately reduce the efficiency of the process. Then they are temperature sensitive. So if we look at the Vidarbha region of Maharashtra, the temperature is comparatively high when we compare it to the north and south Maharashtra. So temperature is also an important part that plays in that. So these microbes are temperature sensitive, and that is why I suggested that using blue green algae would be much better when the area are having high temperature. Then the low cell count is the amount of cells at the particular ml. The cell count is lower in the case of carrier based. Why it is lower? Because since the carrier is involved in the process, the cell is getting attached on that carrier, and ultimately the amount of, the number of cells per that particular into that particular volume is getting reduced. When we look at the liquid based fertilizers, there are several advantages. The first is longer shelf life. Since they are packed, there are very rare chance of contamination. Again, they are in liquid form, so the cell I'm sorry, the cell having moisture around them will definitely have more efficiency. As we know, enzyme need water to carry out the metabolic activities. Similarly, they are temperature tolerant. However, there are several disadvantages such as high cost and high investment. So let's focus on the carrier part. So how could we use this agriculture waste which is being produced as a carrier? So in a two-way we can use this agriculture waste. Either we can use it as a carrier, where the grown microbes are being carried, or are being put on that carrier, and that carrier is being circulated into the market. However, there is another type. We all know that to grow those microbes, we need to provide them nutrients. So what we can do, we can use this food waste or this crop waste as a carbon source, for that energy source for that particular microbe, and let them grow on that. So in that way, the expenses of nutrients which are normally used will be reduced, and that can be efficiently used. Now let's focus on the carrier. So carrier is a medium that can carry the microorganism in a sufficient quantities and keep them viable under specific conditions. So they can so they can be easily supplied to the farmers. And the organic waste from animal production and agriculture food processing industries cause substantial environmental impact. So this waste, what if this waste can be used as a carrier? So to imply that, we must understand what are the properties, what are the ideal properties that should be in a carrier. So the first thing is it should be the non-toxic to the inoculant. So that the microbes which is being loaded on that carrier, that material should be intoxic. Material should be non-toxic to that microbes. If that material is toxic to that microbes, even though we load that microbe on that carrier, eventually it will get killed in the process. Similarly, it should also be non-toxic to the plant on which it is supposed to be applied. So you understand this, the material should be non-toxic, and all the biomaterials, all the biomasses, all the bio waste are non-toxic. We know that. So this criteria can be filled with the biomass carriers. Then the good moisture absorption capacity. We all know the plants have a very good moisture absorbing capacity. And if we can make a small granules of those structures, and we all know lower the size, higher the surface area. As we make the granules or as we crush them down, those small particles can have a larger surface area and can accommodate more number of microbes on them and can efficiently enhance the overall process. Then the third criteria should be, it should be easy to process, and it should be free lump forming materials, so that material should not form lumping. So if you take a biofertilizers and if the process is like that, you have to put that into a 100 ml of 100 liter of water and then apply, and if it starts forming the lumps, then it is not ideal, because that lump will not distribute it equally in the field. Then the another part should be, it should be easy to sterilize. This is very important factor. When we are loading some microbes on particular carrier, we must consider that there should not be a cross contamination. If there is a cross contamination, other microbe is involved in the process, then there is a competitive inhibition between two types of microbes, and that will eventually kill one of them. And what if that one of them is our desired one? So it is very important that it should be easy to sterilize. The material should be easy to sterilize. It should be inexpensive. Of course, this is one of the most important factors. It should be inexpensive. Good adhesion to seeds. So there is a process where biofertilizers can be applied on the seeds, and that seed can be sown to the field. So that application part, where when we apply these biofertilizers on those seeds, that should adhere to those seeds properly. So good adhesion property should be there. Good pH buffering capacity also should be there. Why? Because microbes have a very optimized pH at pH levels at which they can grow. And if the pH is drastically alkaline or acidic, that might hamper the desired culture, microbial culture, and ultimately the biofertilizer efficiency will be lost. And again, as I told you earlier, it should be non-toxic to the plant. So even though these properties are potential or important, crucial to understand, several factors, the ultimate decision should be taken after considering the microbial multiplication level, their surviving during the storage, their general method of planting, and also the equipment used in the process. So all these factors should be considered before concluding to the specific biofertilizer application in particular field. So this is the example, this is the actual studied work where wheat bran and sugarcane bagasse was used for Rhizobium and Aspergillus niger species, where the inoculant was prepared and applied into the solution. So now let's go to the sterilization part. Why sterilization is important, I already have discussed. Let's dive into the sterilization part, how we could attain it. There are two ways of sterilization, as we have seen in the microbiology lab, that most of the sterilization is carried out in an autoclave, which is nothing but a pressure cooker in a simple language, where 120 degrees Celsius, I'm really sorry, 121 degrees Celsius temperature is attained at 15 bar pressure for 60 minutes, where these microbes are 99% of microbes are killed. However, it is to be noted that during the autoclave process, there are several modifications occurs in the basic composition or structure of particular uh, component, which can alter their functionality. And that is why, even though autoclave can be used, using gamma irradiation would be preferred when we are talking about the sterilization of carrier. Why? So this, what gamma irradiation does, it's a it's a process where almost no change in physical and chemical properties of the material are occurred. So since there are no changes, so whatever features you are expecting from that particular carrier, whether it is porous structure or whether it is adsorbity or whether it is absorption capacity, moisture holding capacity, everything would be retained since its structure is not altered. And that is why even though there is an option for the autoclaving, we can use gamma irradiation. But however, gamma irradiation exposure cannot be uh, I won't say cannot be. It is not that feasible at ground level. However, during the production of mass level, we can definitely use this process. Now we have the carrier, which is isolated or which is obtained from a agriculture waste. Now let's understand. Now we have the carrier. Now we have to upload the or we have to load the microbe. Now that microbe which you are loading, that must have some criteria. On that basis, you have decided that you are going to use that. So for that, we must understand there are several parts involved when the screening of such microbes are done at laboratory level. This screening cannot be done at the grassroots level, but the studies carried out in the laboratory can definitely help the ground grassroots level people to employ it. So there are 7 parts involved here. I will briefly explain. The first part is the isolation of microbes, which we are expecting to have that particular trait as a biofertilizers. Then we will screen for that specific criteria. If it is solubilizing the phosphate of phosphorus, we will put a insoluble phosphorus and try to check whether it is solubilizing it. So we will check its state whether it is doing it. Then we will testing under axenic condition. Axenic conditions means we will try to provide a condition which is only favorable to a specific particular single species at the time. So if we do that testing under axenic condition, if that species is there, it will eventually be viable and survive. Then the fourth part is the pot trials and small case testing, where we can try that particular microbe at pot level. Instead of going for the land application, direct crop field application, we have to analyze it at a pilot scale, and that is why we will try to carry out some pot trials and small scale testings. Similarly, then once these part scale trials are successful, then we can go for the trial under the field condition or in situ evaluation. Then once it is successful, we can standardize the studies. So we can put a quality assurance setup on that. So every time we are producing a batch, we would have a exact, not exact, since this is a biological component, it will never have the exact composition, but however, we can try to maintain the consistency. And ultimately, if we are taking it as a startup or we are taking it as a research grant, we can definitely go for the commercialization if the results are favorable. So this was the outline. Let's go the part where biotechnological tools are efficiently employed. So the outline of the production of biofertilizers can be basically divided into 3 parts. The culture selection and maintenance, which we just learned. Okay, even though the culture selection could be in a 2 way. Either you have order it from the MTCC, Chandigarh, or NCL Pune, or either you can isolate and screen it from the soil sample and eventually do the characterization study and then use it. It's all up to us. Then the second part is the culture augmentation and mass multiplication. So since generally the commercial media is used, which is again expensive, we can have another option where this bio waste, this agriculture waste can be used as a source of energy in this field. Of course, the results will be not as efficient as the artificial media, but we are trying to produce something beneficial from the waste. So there would be some consequences to that, and we should be okay with that as well. And the last one is the carrier sterilization, which we already have discussed. So the first part is the culture selection and maintenance. As we have learned in this process, the selection of the culture at the initial level, which we call the primary selection level, can be carried out using this method. However, once if we have a something already established culture that can be obtained from agriculture institutes or MTCC or NCL Pune, so what we can do, we can isolate those microbes. Initially we have to subculture the master culture, let them grow, since they are in a freeze dry form, freeze dry form, they mostly are in a dormant stage and take some time to become completely viable. So this mother culture can be subcultured several times until unless the efficient microbial culture with efficient cell count is available. Okay, now let's keep that isolated microbe aside and let's now focus on the medium part. As we know that since conventionally we were going to put artificial media, we were going to feed that artificial media to those microorganisms. Why if we put a agricultural waste? But is that agriculture waste easily amenable to that microbe? I mean to say, can that microbe directly isolate or they extract their nutrients from that waste? No, sometimes they're not. Sometimes they need a simpler structure. As we all know, the plants are mostly made up of lignin, cellulose, and hemicellulose, and lignin gives the rigidity to the plant. So sometimes the breaking that lignin is not that easy, right? And that is why we have to employ some pre-treatments in the process so that bio waste, that agricultural waste, can be used as a nutrient source for the biofertilizer microbial organisms. So basically there are four types of pre-treatments. The first one is the physical, where then second is chemical, then third one is the physiochemical, and the fourth one is biological. So in physical, of course we are gonna only apply biological, and for the obvious reason that it is efficient and environment friendly and sustainability efficient process. However, we must know that what are the different types. The physical will have the meaning of that particular, like if there is the long huge rigid structure can be milled down, can be broken into small pieces. However, it requires intense energy. You know, microwave ultrasonication irradiation requires intense energy. Microwave processes which are successful at laboratory level cannot be scaled up to the industrial level. Why? They are highly intensive intensive energy processes. So physical is out of the scope in this moment. Chemical, we all are aware that how chemicals are drastically eradicating the nutrients and the good things, good factors from the environment. Then the third one is a physiochemical, where chemical and physical methods are combined. These methods are highly efficient, let me tell you that. But however, they again require high amount of energy cost and sophisticated machineries. And the ultimate is biological. So let's assume that whatever, if we apply a complete green approach, then even though our productivity will be not as efficient as when other processes are employed, but it will definitely sustain the environment. And in today's era, the most important thing is to make this planet sustainable. So in biological, there are two types of things: wholesale pretreatment, which is also known as wholesale biocatalysis, and the another is enzymatic treatment, which is known as biocatalysis. So what is the basic difference between wholesale pretreatment and enzymatic pretreatment? So in wholesale biocatalysis or in wholesale pretreatment, what we do, we put that particular substrate, like it is a, like let's assume it's a rice husk. We put that rice husk in a water where we have provided a trace element nutrients. We put the microbe and let that microbe break that rice husk. So the whole microbe is involved in the process. However, it is producing the enzyme in between, but the whole microbe is involved in the process. The process is known as wholesale biocatalysis. The whole cell is involved in the process. But however, the enzymes are being expressed and doing their work. So they are doing the biocatalysis. So that is why it is known as wholesale biocatalysis. Similarly, now you can easily understand what is enzymatic catalysis, where either the enzyme is being purified in a crude form or a completely purified form and then employed. And you can easily analyze that which form would be cheaper. Of course, the wholesale biocatalysis, because the downstream processing of enzymes costs more than 60 to 70% of the overall cost. And when you are using wholesale biocatalysis system, you are reducing the cost by almost 60 to 70%. So these biological processes can be employed for that particular biomass pre-treatment, and that pre-treated biomass now can be used as a carrier or as a nutrient source for that particular microbe which is being used in that particular biofertilizer. The second part is the culture augmentation and mass multiplication. So now you have the strain, you have the mother culture, you have a subculture it several times. So now it has a almost 95%, more than 95% viability. And now what is the next part, that either you can go for the flask level at a primary stage, and if you are doing the mass production, then you must employ fermenters. Those who are working in downstream processing or those who are working in bioprocess engineering, they must be knowing that fermentations, for to carry out the fermentation, if you employ the fermenters with which are sophisticated with equipments, the productivity is high productivity and high yield is easily attained. However, the process is costly. So if we have the both options, either we can use the flask method or either we can use the fermenter method. So at primary level, we can use the mask. At secondary level, we can use the fermenter. And as you can see, the complex organic waste, all the waste, they have protein, they have carbohydrate, and they have lipids. And these proteins, carbohydrate, and lipids can be hydrolyzed to obtain amino acids, sugars, and long-chain fatty acids, which can be used as an energy source for that particular microbe. So even though when we add glucose or when we add jaggery or when we add sugar molecules, we add um, sugar entities, every time that molecule is being used as a simple sugar complex molecule by that particular microorganism. So they ultimately need energy. And if that energy can be obtained with this agriculture waste, that would be really beneficial. However, once the media is ready, now you have a media, you have agriculture waste as a source of carbon. You also have the microbe which you have screened. However, the proportion of media components, the optimization process is very important. Randomly putting something will give you results, but however, if you optimize it, you will definitely get better results. There are several methods to optimize the media. If n number of more than 3 or more than 5 or 7 types of parameters are involved in the process, that time we can employ another biotechnological tool known as response surface methodology. I hope some of you must have heard of this technique. What I'll briefly tell you what we do in that. This response surface methodology is a computational program where we have to put a different parameters with a specific range. And this different parameter with a specific range is being computed, and a programmed experimental work is offered to the researcher. So the researcher has to carry around 17 experiments, as I said, per se. So this 17 experiment he or she has to carry in the laboratory, and the results obtained from these 17 experiments should be again put back to that software, and that software understanding the correlation between different parameters and the output results, it obtains a regression model, and that regression model can give you a formula where you can literally predict the particular output of that particular process by changing the numbers. Even though this sound complex, just try to understand what is the basic fundamentals. Is that we're trying to optimize the media to enhance the efficiency of the process. Okay, if this is very interesting process, response surface methodology, and I hope those who have done would have definitely would agree with me. Then once the media is formulated and sterilized, it is inoculated using the shorter cultures multiplied in the flask. So now we have the culture, now we have the microbe, now we have the optimized media, now we have to mass culture, right? Multiple batches. So this bacterium growth medium is called broth, and this broth is then multiply cultivated or subcultured in several flasks. So we can have a mass culture. After about 3 to 4 days of fermentation period, the broth will be ready for packing in a carrier material. Now we have a carrier material which we had kept aside. That carrier material can be taken, sterilized. Of course, we have to sterilize that, and then this material can be loaded. In case of bacteria, it can take 3 to 4 days, but in case of fungal, if we all know that it will take more than a week or so. So since fungal strain take a longer time to grow, the whole process will be comparatively longer. Again, at various stages, the quality should be tested by drawing the samples. Like we have to take the um, uh, aliquots at a particular time of interval, and we have to check the viability of the cell to maintain the consistency in the process. So there are, when we go for the mass cultivation, there are several types of processes involved, uh, among which the four are the widely known. The first one is the cemented tank method. The another one is the shallow metal through method. Then the third one is the polythene line pit method. These are actually different methods to mass cultivate it. Okay, whether you're doing it since these carriers are being employed in the process, these are again mass cultivated at this level. And there is another field method. Field method is again like a controlled region where the same thing can be carried out. However, most of the people would prefer this kind of things to maintain the uniformity and consistency of the material, so there won't be any other contamination from the surrounding. Then the carrier stabilization can be sterilization. This part is already been discussed. So this material, this bio waste material which you are using as a carrier material, which is whether it must be a crop residue or it must be a dry fruit waste, whatever it is, we can crush it down. Okay, we can form a, again the logic is the same. Higher the smaller the size, higher the surface area, higher the mass transfer. Okay, so smaller the size, the more number of bacteria or the more number of microbes will get loaded on the sample. So minimum amount of sample will have large maximum number of microbes in that form. So if I have to put this flow chart, this whole production flow chart, how this goes. Okay, the first is the mother culture. Then mother culture is taken and slant is obtained, and this slant is subcultured and broth is obtained. Initially, sorry, the initially broth is cultivated in a small flask, and then this small flask can be transferred to the big flask, because scaling up is a gradual process. You can't just take it to the next level. We have to go gradually to accommodate the environment for that particular microbe. Then shakers can be employed. So what shaker does, they shake the flask by providing by providing more amount of dissolved oxygen, and that is why. And this also the shaking also enhance the mass transfer rate. Okay, so these both factors we can employ shakers in some cases if we have the facility. Then the broth is blended with sterilized carrier. Now we have the carrier, which is again, it should be sterilized. This can be blended, and this blended can be, okay, one more thing which I missed here. This broth, either we as we said, either we can go for the primary cultivation where we go with the flask, or either we can go for the large production where we go for the fermenter. So when the broth is blended with the sterile carrier, this can be go for the packaging in polythene bags, similar to as shown in the last slide, and that can be stored at 25 degrees Celsius and can be dispatched to farmers whenever is required. So now we have understood that how biotechnology can be employed, how waste can be used. Now let's understand what are the critical challenges, what are the issues that generally faced by farmers in this regard. Before that, let's try to understand, once this material is ready, how we are going to apply it. There are three types to apply this biofertilizer. The first one is the seed treatment, the second one is the soil treatment, and third one is the seedling root deep treatment. So let's go one by one. What happens in seed treatment? This is the most common practice in applying biofertilizer. So what they do generally, they take the biofertilizers, if it is in a gel form or it is in a slightly liquid form, or sometimes it is in a solid form which can be added into the water and the solution can be obtained. And that solution, these seeds can be dipped into that solution. So that seed will uh, have those microbes around its shell. However, the adhesion capacity of carrier should be good. Otherwise, only dipping won't help. It should get adhered to that structure, seed structure. In this method, the biofertilizers are mixed with 10% solution of jaggery. So jaggery again sometimes help as a sugar source again. This slurry is then poured over the seeds. Either the slurry can be spread over the seeds, or seed can be dipped into that same thing. The treated seeds should be dried in the shade. So these seeds now should be dried. But during this drying, we have to be careful that it should not get exposed to the higher temperature. Otherwise, the structure of those microbes will get denatured. Generally, 750 grams of biofertilizer is required to treat the legume seeds that can be applied in a 1 hectare area. Other essential criteria is that when seeds are involved, the seeds are not always sown immediately. So maybe seeds are dipped into fertilizers and kept it in the storage for a longer time. So in that duration, the bacteria have to survive, or that fungal strain has to survive. Otherwise, its efficiency at the end will be very less. Then the survival of the inoculant bacteria during the storage period, the same thing. Similarly, when they are applied to the soil, it should not harm the soil. These things we have already discussed as an ideal characteristics of carrier. Let's go to the next part, which is soil treatment. So this method is mostly used for fruit crops, sugarcane, okay, mostly fruit crops, where localized application is needed. So we put that fertilizers near the plant root. At the time, so sometimes the biofertilizer are also broadcasted, like sprayed all over the plants, but may require 10 times more biofertilizer. Obviously, if you spread it just like that, the most of the biofertilizer will get wasted in the process and should be applied accordingly. Sometimes it can be also added with the farmyard manure, and then so this farmyard manure can be mixed with this biofertilizers. So there are two things happening here. The equal distribution of biofertilizers can be attained. Apart from that, manure, which is farmyard manure, will help as a potential platform to survive those microbes, so they can efficiently start proliferating in that surrounding. And if they are proliferating in a more amount, of course they would eventually helpful to the plant. Then the third part is a seedling root tip. So what they do, they take a seedling. Instead of taking a seed, the seedling is a small plant which is grown from a seed. That particular small seedling is taken and dipped into that fertilizing solution, biofertilizing solution, and can be used. This is mostly used for the potato tubers. Then now let's understand the critical challenges. How could one get good response to biofertilizer application? Okay, the first thing is the biofertilizer product must contain good effective strain in appropriate population. The strain is efficient, but the population is less. So the soil is a steady thing. The microbes has to circulate themselves. So that is why they should be in an adequate amount of population, and they should be efficient as well. A right combination of biofertilizers should be used, and it is also suggested that the appropriate time should be considered for that particular application of that particular microbes. For seed treatment, adequate adhesive should be employed. As again, if you are applying the biofertilizer on the seed, the adhesion should be better for the carrier molecule. Otherwise, it would not stick to the seed and eventually get washed off in the process. Also, ensure the supply of phosphorus and other nutrients. So this, whenever you are selecting a biofertilizers for the particular plant, we must understand the distribution of nutrients as well. We can't just focus on a single nutrient and keep putting that biofertilizers for that plant. Then what would be the probable reason for not getting a response from biofertilizer? This might happen in the actual field, that people are exploring their biofertilizers in the field. However, they are not getting any response for the same. So in that case, what can be done is that we have to sit at different level. On account of quality of product, let's see if the strain is ineffective or not. Whether insufficient population of microorganism is there. If it is insufficient, we can put some more. We have to check whether it is cross-contamination of particular microbe. Like we put some particular biofertilizers and some other microbe is present in a large amount there, that can of course have the competitive inhibition and ultimately kill the desired microbes. On the account of inadequate storage facility, we must consider that it should not be exposed to the higher temperature when it is stored. We should also see that the storage unit is sterilized and cannot promote the cross contamination. On the account of uses, now how it is being used should also be considered. It should be put in an appropriate dosage. Poor quality adhesive, again the same point. Strong doses, excessive doses can also hamper things. As we all know, this biological system should be balanced. Anything which is in excess would damage the ecosystem. On the account of soil and environment, high soil temperature and low soil temperature. If the temperature is high and the microbes which is being used in the biofertilizer is temperature sensitive, again it will definitely hit that. Then the acidity and alkalinity of the soil again matters. Again, the pH of the surrounding again matters for that particular microbe as well. Then there are other factors as well. Let's go to the next part. What precautions one should take for using biofertilizers? First, they should be stored in the cool, dry place. They should be uh, they should be highly isolated from the contamination chances. Right combination of biofertilizers should be used. Other chemicals should not be mixed with the several times what they do, they take some farmers take chemical fertilizers and they put with the microbial fertilizers, but they eventually forget in the process that these chemicals are literally killing those microbes in the process, and that is again the efficiency will be lost in the process. So biofertilizers are live products and require care in storage. Even though they look not moving, these are live microbes and we have to store them accordingly, we have to conserve them accordingly. Then let's now talk about the advantages. As we have already discussed the advantages, let's briefly go into this. The association with other soil microbes, which is very important. What biofertilizers do, they attain the microflora enrichment. So they get associated with the surrounding and they get this symbiotic association which is naturally occurring and ultimately help the whole thing grow. Plants are also growing, fungus are also growing. This is how the system is getting sustainable. They fixes the atmospheric nitrogen. This is a very important part. They are fixing the atmospheric nitrogen. They also stimulate the plant growth while improving the soil properties. They enhance the biofertility and they are causing no damage to the environment. These are the again the same advantages which we already have discussed. Now let's go to the disadvantages. As such, there are no harmful impacts of the biofertilizers. However, they are sometimes very specific to particular plants. So some biofertilizers would not help particular species of plant, but will help the another one. So this is not disadvantage, but a little setback. The acceptability of biofertilizers has been rather low because they do not produce quick and spectacular response. So it is like Ayurveda. They take some time. Even though they are efficient, they take some time. And people are running out of time. They are in hurry. They want everything to happen overnight. And that is not going to happen in that case. Biofertilizers will take their time, their natural time. But eventually, in the long run, they will be very, very helpful. Again, this requires skills in the production and application, and again the storage part is another hurdle. So before let me see the time. Okay, before okay, let's go to the next part, which is a case study I have. This is 2-slide session where I would try to put a study where what they have done is that they have used phosphate solubilizing microorganism that can transfer insoluble phosphate to soluble form using agricultural waste as a source, as a carrier as well as a source. In this study, Aspergillus niger, which is a fungal strain, was isolated and identified as an efficient PSF microbe. The strain generated 689 milligram of soluble phosphate per liter in NBRIP media, which is a type of media, after 10 days of culture. To produce an affordable biofertilizer using Aspergillus niger, the potential of widely available carrier materials for growth and maintenance of the strain were evaluated. So what they have used different kinds of waste, like whether it is corn cob, whether it is wheat husk, or whether it is composted cattle manure, that was used as a carrier. So what they are doing is they are using the agricultural waste as a carrier. So what they found is that in the first 5 to 6 months of storage, the carrier sterilized by gamma ray irradiation maintained high inoculum loads. So the shelf life was almost 100% reserved till the 6 months. However, after 6 months, it start decline. Since these are the biomaterials, they will start getting decomposed eventually and ultimately. But however, 6 months is a quite acceptable shelf life. When this biofertilizer was applied to the Chinese cabbage in a pot experiment, so since this experiment was carried by an academic institute, it was carried in a laboratory, and when the biofertilizer was applied to the Chinese cabbage, there were several number of Aspergillus niger spores were found, and the fertility of the soil was drastically enhanced. So apart from this structure, there is another approach which can be also used along with these approaches, is the introduction of mixed inoculum. Like, as we know, in this natural microflora, the microbes, these constituents of different microbes are working together. So why not find this kind of combination in the laboratory? And if those combinations are working out, why not use them efficiently? So this is another approach which should be kept in mind while trying to screen an efficient type of biofertilizers. So the next part is biopesticides, but I will only go to the biopesticide basics because the fundamentals is the same, that we are going to use microbial uh, biopesticides specifically. Even though there are different types, we will only look for the microbial pesticides, because microbial pesticides can be fed up with the agricultural waste as an energy source, not others. Let's go into that. So let's see the potential of biopesticides, that more than 150,000 tons of pesticides are used in agriculture in India annually. But these are the chemical pesticides, not natural pesticides or biopesticides. So now you can imagine how much pollution is being created through this process. Because when pesticides are applied, like they apply the pesticide on over the field. However, only pesticides which is there on the plant will be affected, but simultaneously the other friendly microbes or friendly insects, again there are some friendly insects and friendly microbes, those are having symbiotic association with that particular crop, will also get killed in the process. And that is why eradicating or eliminating the commercial or chemical pesticide is very important. And pesticide can be replaced with the biopesticide. Of course, there is an option. The intensive use of chemical pesticides in agriculture is causing a serious concern. The problem is especially serious because of the development of resistance of pesticides. So what happens, that farmers start spreading the pesticides this year, second year, and then the year after that. So what is happening in between, that the pests which are there present, they start developing their resistance power against that particular pesticide, and we keep putting that pesticide on the ground. And those pesticides, those are targeted pesticides, have already obtained their resistance against that biopesticides. But the others which were not the target in the first place will getting affected. They're getting eliminated from the ecosystem. The ecosystem is getting imbalanced, and that is why using a biopesticides is very important. So we all know what are the advantages of using biopesticides. Again, they are derived from animals or sometimes plants or microorganisms. So even though they are derived from animal, plants, and microorganisms, we are in this aspect, in this regard, in this perception of applying biotechnology in the field of biofertilizer, biopesticide, we will mainly focus on the microorganism biopesticides, because microorganism biopesticides can be, because my growing microorganism needs nutrients, and that nutrients can be fed with the agriculture waste. So what are the advantages? That they are more target specific than chemical pesticide. They are very specific. When you look into the types of biopesticides, you will understand that they are very specific, because they are natural. They are actually not pesticides. They are just carrying out their natural phenomenon. They are killing their host, but eventually their host is our enemy. Okay, so that is why they are very specific. They won't eat any host. They have very specificity for that, and that is why these biopesticides are highly specific. So let's dive into different 5 types of pesticides. The first one is the Bacillus species, which is a type of bacteria. So this is a microorganism based biopesticides. So what it does now, it releases some kind of toxin. Okay, the toxin can kill this um, pests. This pest, the toxin can kill pest. This pest. So what researchers have done, they have isolated that from that bacteria. They have isolated a gene which is responsible for that toxin secretion and added to the plant itself. So the genetically modified plant is also there. Okay, so the genetically modified plant, when that insect or that pest eats the leaf of that particular plant, since that leaf has that toxin, it will eventually get killed. But however, using again, I'm going back to the my earlier point that using genetically modified plant is a getting approval is a lengthy process, and eventually there are several ethical issues as well. So, so we can use this bacteria as a source of biopesticides. And for the growth of these biopesticides, again, by microbes, we can use agriculture waste as a source of energy. There is another type which is known as baculovirus. Okay. So what this baculovirus does is, we all know what viruses they do. They literally kill the host cell. Okay, most of the time there are lysogenic and lytic cycles. I will not go into that, but they are very lethal. They are brutal. They kill the host cell. So baculoviruses are the target-specific viruses which can infect and destroy the number of important plant pests. So these viruses can be spread on the pest, and when those viruses are get entry into the that pest body, eventually that um, harmful pest will die in the process. The neem plant, we all aware of this. There are a number of plants, but neem plant has um, say highly valuable. It has been established as a highly valuable pesticides since it has azadirachtin element, which kills the pest, and that is why you will rarely find any infection to the neem plant. You would rarely find any pests or any infection to the neem plant. This is the list of several types of plants which can be used as a biopesticides. However, the next part is the fungal one. So bacterial one, the fungal one, the trichoderma is a fungal species which can be used again several soil-borne diseases. Soil-borne diseases are the diseases those are caused by the microflora or the microbes which are present in the soil. So every microbe present in the soil should not be a friendly association with the plant. Some would be having a enemy association as well. So in that cases, this fungal strain can be used as a biopesticide and applied in the field to eradicate those kind of pests. Then this is another type of this is just for the information. That's trichogramma the insect named insect. What it does, it lays its eggs into the pest nest or pest with the pest eggs. So whenever those wasps, this is a type of wasp, insect, but a wasp. This wasp, when they come out from the egg, they start eating the eggs which are just residing there. So what is that? This is a really good thing, but this is a natural phenomena. Okay, so employing this is not that feasible as you employing bacterial and fungal species, but this is really good because the damage control is occurred before the damage actually occurs. So the eggs are getting killed in the process, so there won't be any pest formation, there won't be any ultimate damage to the plant. So in the beginning itself, the pests are getting eradicated from the system. So this is very efficient but not that feasible. However, the other parts are feasible. So now let's conclude the whole thing, how government policies are helping this adopt wider adoption of biofertilization is taking place. Let's last 3-4 slides and I'll wind it up. So despite the Indian government efforts to promote the production and the use of biofertilizers, various studies found that they have found little acceptance among farmers. There is a reason for that. Even though we all understand that organic farming is the future, we all understand that organic farming is the only way to make this planet sustainable. Why we are not adopting it? So there are several problems. Let's see one by one. The present policy of providing grants to the low interest loan to the biofertilizer producers, so anybody can attain a loan. So this has good and bad impact as well. So what is happening is that the quality of the biofertilizers which is being produced is drastically getting affected. Okay, that is why no quality biofertilizers are there in the market, and that is why if there is no quality biofertilizers in the market, there won't be any implementation or adoption of wider adoption of that biofertilizers. Apart from that, the policy of marketing biofertilizer at a very low price is also not good, because when something is sold at a very cheap price, selling at a cheap price is a good thing. But if you want to sustain a system or sustain an industry, selling something at an extremely cheap price will not attract the investment. And if there is no investment, there won't be a propagation or industrialization of that particular field. And if you want to cater the demand, the huge demand of Indian agriculture sector, we must have the industrialization of this field, which is not yet been attained. Again, the storage is another concern which should be addressed. Research and development to identify more sustainable strains. So academic wise, what we can focus our research towards a direction where we can either we can isolate or either we can genetically modified microorganism strains which are highly efficient and highly targeted to that um, particular purpose of biofertilizers or biopesticides. That can be done, that can be done in the laboratory, and if successful can be taken into the plant level and ultimately scaled up to the industrial level. The present government policy of promoting biofertilizers without ensuring good quality and performance has actually harmed their cause, because even though they are providing the grants, they are not monitoring the quality. There is no quality assurance of that. So even the people are getting the grants and start putting the plants, the fertilizers does not has that quality which is expected, which is anticipated. If the anticipated quality is not there, ultimately it will never find a wider adoption. Then similarly for the biopesticides, intercropping should be the focus on sustainable agriculture by promoting intercropping as well as reduce crop intensities. So we have to put some gap between one yield to another, and we should also put plants or use varieties which are pest resistant or resist resistant or sometimes drought resistance. The improve the intensity of training and integrated pest management. So the pest management is the integrated pest management is a program where several types of activities can be combined to attain a long run pest control in particular field. That should be attained. Also, the promotion of the use of pesticides by the state agriculture departments and IPM workers should be carried out. So there is a lack of awareness as well. So in these tribal areas, they of course they do have their own way of farming, but it doesn't mean the way of farming they are doing must be efficient. There must be some other way, but they are not aware. So this awareness can be spread by the government employees as well as government departments as well as IPM workers. And the state agriculture universities can also promote the research in this regard, and we can have decisive influence on the overall agriculture sector. So with this slide, I will wind up my lecture, and I hope this lecture was slightly helpful to all of you, and thank you for being patient for such a long time. Thank you so much. Thank you.
Sir, if it is okay for you, we would request our attendees in case if they have any queries.
Please go ahead with the questions. If you have any queries or if you have any suggestion you would like to discuss with others, I'm open to that. We are all open to that.
Very good evening, sir. Am I audible, sir?
Yes, you're audible. Audible, sir.
This is Dr. Arun, sir.
Hello, sir. Hello, good evening.
Good evening, sir. Sir, actually, your presentation was excellent. The PowerPoint presentation was very much impressive.
Thank you so much.
And I do have some questions regarding the culture preparation, mass culture preparation, sir.
Okay.
Shall I, sir?
Yeah, please go ahead.
Okay. Sir, you have told that the fermentation period around 3 to 4 days for the mass culture of the culture preparation. Yes, sir.
For
For
bacteria, yes.
bacteria. So after that
Yeah.
particular fermentation process, right? Uh, is there any idea to see the biomass growth in that particular bacteria, sir? Any methodology is there, sir?
So what we can do, if you have worked on a bio fermenter, you must be knowing that
Yes,
after
sir.
a particular time of interval, we have to analyze the viability cell count, and if you are looking
Yes.
for a particular expression of enzyme, those
Yes, sir.
aggregates can be taken out and can be analyzed with the UV spectroscopy, and that
Okay.
result, the cell count as well as the viability of that particular strain can be maintained. And this is
Okay.
how they actually do in the laboratory when fermentations are carried out. They have to see, or they have to regulate the consistency of the strain, and that is why
Okay.
this whole growth curve kinetics is being followed. You must have heard this term, growth curve kinetics. And these growth
Okay.
curve kinetics are, yeah, efficiently being employed in the process to obtain or to remove the culture at its best efficiency as possible.
Thank you, sir. My next question is, after the fermentation period is over, is it necessary to follow the downstream processing, sir, like purification
No,
part?
no,
Or,
not necessary, sir. Not
okay.
actually, it depends. Now, just give me a minute and I will explain.
Okay.
So it depends whether your product is what kind of product you are expecting. It is all depends on that. Wholesale biocatalysis has their advantages, but also has their own disadvantages. Is that the culture will have the different types of, when microbes are grown in a solution, there are
Yes.
several types of biological factors are produced. So there won't be that enzyme, that particular enzyme will be in the solution, but not necessarily the other components which are present in the solution might uh, interact or might uh, hamper the overall efficiency of the process. And that is why in those cases we are go for the purified culture, and the downstream processing is causing a lot of cost, so it's not necessary that we have to use it. But if this reaction is getting hampered by the other components present in the solution, in that case we must express and purify the enzyme and then use it as in a crude form or in a purified form, or we can also go for the immobilization process and use it.
Okay, sir. Okay, sir. Thank you, sir. And my last question is,
Yes.
you have told that it has to be stored in the cold conditions, sir.
Yes,
So, what is the
but
necessity
25
of
degrees Celsius.
25
Yeah,
degrees, sir? And again, before submitting to the farmers, it must be around 4 degrees centigrade. Am I right, sir?
yes.
So, what is the reason, sir? Okay, if it is
So,
before reaching to the
so
farmer,
I will tell you. I will tell you. Is your question done, sir?
yes, sir. What is the reason for storing the 25
Yeah,
degrees
okay.
centigrade and 4
I'll
degrees
tell you.
centigrade?
I'll tell you. So what happens is that when the microbes, the temperature is reduced, the metabolic activities of that particular microbes are slower down. They are
Okay.
not stopped but slowed down. So this slowing down is important. If microbes keep growing, like we are trying to use a carrier which is a agricultural waste, okay,
Okay. Oh,
so
okay.
if
Okay.
microbes start growing on that carrier, ultimately the carrier will be consumed in the process, and the number of microbes will be increased, and it
Increase.
might happen that the whole structure is getting destructed in the process. And that is why it is very essential that we reduce or we slower down the metabolic activities of that microbes, and that is why putting them in a cold storage is important.
Thank you so much, sir. Thank you so much. Thank you.
Welcome, sir. Welcome.
The attendees are requested, in case if anybody else has any queries, you can just unmute yourself and ask your query. If you are not comfortable unmuting yourself, you can also type in your query, or you can share your experience in the chat box as well, and our expert speaker can just read it out and answer it. So if there are any queries, please go ahead. Please feel free to ask.
Thank you, Dr. Arun. Thank you so much.
Sir, sometimes it takes time for them to type in the chat box, so we can
No
catch
worries, sir.
it
No
from there.
worries. No worries. No worries. Thank you, Srivashini, for appreciating the presentation. Thank you so much. Thanks a lot. Thank you for being patient, everyone. Thank you so much.
Sir, this is a layman question.
Yeah.
However, I always had such queries because I'm not from the biotechnology branch, but I do have some
Yes,
interest
yes.
in,
No, no
yeah,
worries, sir. No, please
in
go ahead.
developing, like what can we say? Like I have this hobby of gardening. So I wanted
Mhm.
to know, sir, there is this methodology of which I have heard they say that it is one of the ways of growing forests really fast or growing plants really fast. And they call it as something Miyawaki forest or something like that. Miyawaki afforestation. So is it also a natural method of growing trees and plants fast and
Sir, to be very honest, I am not aware of this technique. Since plant biotechnology is just a topic which I have learned, this technique I am not aware of. So I will not able to comment
Okay.
on that.
Okay. Actually, I saw that video on. I keep watching those videos, and I saw that video on
Okay.
YouTube.
It seems you are very interested in gardening and all. And if you could text me, and I will definitely send you some information on that.
Sure, sir. Sure. Yes, sir. I will do that.
Okay.
Okay, so I suppose that there are no further questions from the attendees. So, sir, on behalf of Ballarpur Institute of Technology, Ballarpur, and on behalf of organizing committee of this short-term training program, I would like to extend our heartfelt gratitude and many thanks to you for conducting such a wonderful session. Thank you for accepting our invitation on such a short notice. And at the same time, the session was really what can I say? I am myself not from a biotechnology background, and yet I found it really interesting. And the information was so detailed. I think that you took us through every step, so that in case if somebody wishes to practice this methodology, then I suppose right from listening to your lecture, it could be very easily possible for them. So thank you, sir. Thank you so much for conducting such a wonderful session for us.
Thank you. Thank you so much, sir. I sincerely thank you, Dr. Mishra and Abhijit sir, for inviting me for this STTP program. I'm honored, and this was a very wonderful experience. And I'm looking forward to the future associations as well. Thank you so much. Thanks a lot. And I also would like to thank all the audience for being so patient and being so cooperative throughout the whole session. Appreciate that. Thank you so much.
Thank you, sir. Now, to all the attendees, you will receive a feedback link on the WhatsApp group and to share your feedback for this particular session. And you can fill in the feedback form through accessing that link or through the WhatsApp from the WhatsApp group. The link will remain open till 11 p.m. tonight, so please submit your responses before 11 p.m. tonight. In case if you have any doubts or any queries, you can always get in touch with the coordinator of this STTP, Dr. Archana Nimkar ma'am, or co-coordinator of this STTP, Professor Neeraj Singh Best sir. Having said that, we would declare that the session, today's session is concluded here. See you all tomorrow at 12:30 p.m. Our tomorrow's STTP is scheduled as usual at 12:30 p.m. So we will request you to please join us by 12:15 p.m. so that we will be able to start the STTP at sharp 12:30 p.m. as scheduled. There is also a small announcement that we do have best participant award. We have already mentioned this earlier as well. So in case if you are your attendance is regular, you are interactive during the sessions, and at the same time if the speakers, if they appreciate your questions and things like that, then automatically you are nominated for the best participant award. So all the best to all of you, and make sure that all of us we make the most of it through this STTP. Having said that, we will conclude here. Thank you very much.
Thank you so much.